You ever wondered when you take a bite of food how your body knows or is able to get all those sugars and the nutrients in the food that we eat to all the cells of our body? Or in that fact, why for the most part does everyone have 5 fingers in the same exact sequence? I'll tell you, it's cell communication. In the yeast Saccharomyces cerevisiae, there are 2 different forms of the yeast cells. There's an A cell and an alpha cell. A cells secrete A factor, alpha cells secrete alpha factor. When alpha factor binds to an A cell, or when A factor binds to an alpha cell, it causes a chemical reaction or response within that cell to start growing towards the source of the signaling. Once these chemical molecules bind to the cells, it triggers a cell response for the cells to start growing towards each other, and eventually they fuse. And when they fuse their genetic material, allowing for a blend of the genes that are contained in each original cell, and then through a series of divisions they end up with offspring cells that have genetic diversity from the original parent. So the question is, how does receiving a signal on the cell membrane lead to a cellular response like the growing of the cells towards each other? So to answer this question, we can look at the big picture of cell communication, and it can be broken into 3 different stages: reception, signal transduction, which is converting that received signal into a biochemical pathway, and then finally cell response, so how the cell will respond to the initial stimuli that is received. Reception begins when the target cell detects the signaling molecule. To detect the signaling molecule, it physically binds to the membrane protein receptor on the exterior or of the plasma membrane, or in some cases, like for signaling molecules composed of lipids, they're able to pass through the membrane and bind to receptor proteins inside the cell. It's important to remember that the plasma membrane is a mosaic of protein receptors that all have unique 3D structures based on their amino acid sequence, and those 3D structures allow them to be specific or complementary to specific ligands or substrates that they'll bind. So this means that receptor proteins are only going to detect specific signaling molecules. Once the signal is detected, meaning the signaling molecule has bound to the receptor protein, this causes a change in the receptor protein. This change activates a series of biochemical pathways that change that chemical signal into a cellular response. These biochemical pathways are oftentimes a series of many steps, and each step is facilitated by a relay molecule that gets activated by the previous step. Ultimately, the final relay molecules will activate this cell response. Cell response can include activation of enzymes that already exist in the cytosol of the cell, causing them to go facilitate some sort of metabolic process perhaps. A second possible cell response can be causing alteration in the cytoskeleton, which is going to change the shape and structure of the cell. Or this transduction pathway can activate some transcription factors that turn on genes that lead to the production of the proteins they code for, and those proteins would proceed to complete cellular work. Because of the specificity of protein receptors, each signal transduction pathway leads to a cell response that deals directly with responding to the initial stimulus. Now that we've examined a general overview of how cell communication works, let's take a look at the variety of forms that these signaling molecules can take. To begin with, some signals don't travel very far, and these are called local signaling molecules. And then other signaling molecules are able to travel much farther distances, and these are called long-distance signaling molecules. One type of local signaling molecule can occur when you have animal cells with gap junctions or plant cells with plasmodesmata. Those are just both examples of where you have the membrane or cell wall of the plant physically touching and there's shared fluid between the cells. So any signaling molecules that are dissolved in the cytosol are able to travel across to other cells and be received by proteins, causing a cell response. Other signaling molecules might travel a bit further, but still not as far as long-distance signaling molecules, and it would only affect cells in their local vicinity. Examples of this would be paracrine signaling molecules, like growth factors, that their secretion during growth development, especially in the embryo, is what leads to specific changes in cell division at coordinated times. And it's the concentration and timing of these that leads to a lot of the important changes that occur in our body during development. Local signaling is very important for maintaining homeostasis and regulating cell function. However, it doesn't work alone. It also is highly dependent on long-distance signaling. Long-distance signaling molecules, often referred to as hormones, are very important in both animals and plants. In animals, hormone signaling is often referred to as endocrine signaling because the endocrine system is composed of specialized glands and tissues in our body that produce and secrete signaling molecules into our bloodstream. Once in the circulatory system, these hormones travel around to all the different tissues and organs in our body and are able to cause cellular responses to any cells with receptors to that specific signaling molecule. For instance, insulin is a signaling molecule released by the pancreas in response to high blood glucose levels. This signaling molecule can target our body cells, causing the uptake of glucose into the cells to be used for cellular respiration to generate ATP, or if those signaling molecules target cells of the liver, it can cause uptake of glucose and the polymerization of glucose into glycogen for long-term storage. Here we can see how variation in transduction pathways can lead to different cellular responses. Either absorption of glucose by our body cells to generate ATP through cell respiration, or the storage of the glucose as glycogen in the liver. Plant hormones, often referred to as plant growth regulators, can either be transported through the vessels of the plant tissues, or oftentimes they simply diffuse through the cells or the air to their targets. For instance, ethylene gas is a plant signaling molecule that diffuses through the air, and upon its detection on the surface of the cells leads to fruit ripening or growth. That's why oftentimes they make the suggestion to put your fruit in a paper bag so it builds up the concentration of ethylene gas within the bag, promoting fruit ripening, allowing your fruit to ripen a little bit more rapidly than if they were out in the air. As we wrap up our overview of cell communication, remember there's 3 stages of this process. Reception, where the signaling molecule binds to a specific protein receptor either on the plasma membrane of the target cell or inside the cell. Signal transduction, the conversion of that signal that's received to a biochemical pathway, leading ultimately to cell response.